what was really the inspiration for the development of this platform, and what biological questions does it really help address that traditional 2D cultures or spheroids cannot really necessarily as well?
A tumor inside the human body is a clump of cells that are interconnected. They communicate with each other. They are connected through an extracellular matrix. Think of it as a mesh, and that mesh also communicates with them, and the tumor communicates with the other physiology or the other organs. So it's a very complex system, and these cells somehow they evolve under stress, like us human beings. You stress us, we become more active, more willing to survive, more, how can I say, we evolve under pressure. So a complex tumor is under a lot of stresses, similar to oxygen, for example. So when under stress, the response to drugs could be could be different. So you would think that a certain dose would help kill a certain tumor, and rather killing it, putting it under the perfect stress conditions, that it would even grow faster. So dosage and testing at the preclinical stage is very important to mimic exactly the real complexity of that tumor inside the human body. So that's why cells that are grown in a petri dish, they are relaxed in two-dimensional frame. When you dose them with drugs, they quickly die. But ones that are evolved under a lot of stress, of less oxygen, less nutrients, these cells are monstrous. So I'm talking in a language, in a layman language that everybody speaks. I'm trying to stay far away from the technical aspect of it. So these cells, they become monstrous, and their resistance to drug increases. So basically what we are trying to do to solve this is to mimic that complexity for our drugs to fail less, basically.